production of the graduate students, allowing the exchange of ideas. I'm sorry, I have something here. The exchange of ideas, the best scientific production, the innovation, and also scientific development. This type of seminars and events accomplish our main missions at the faculty and at the university, the mission of developing scientific research, of investing in the quality of teaching, graduate teaching, and also the opportunity to exchange with international institutions and prestigious universities like the Sydney University. It will be also an opportunity, a huge opportunity for the graduate students to exchange results, developments, ideas with their peers at University of Sydney. So I express my joy for this and for being here, and want to thank the organizers at the University of Sydney and at the University of Porto, hoping that this will be an opportunity for further work and cooperation, and wish you all a productive and a pleasant seminar. So thank you for this opportunity to open this joint seminar with you. And thank you all for thank you. So Fernando.
Thank you so much for the introduction. Now, Rita, I'm not sure if you want to take things on your end and.
Thank you, Fernando. I might pass to Mayuresh. He's the co-director of the Brain Dynamics Center, and he might have some words to say to introduce us.
Sorry, are you able to see that screen? Yes. Firstly, thank you so much for that warm welcome, Professor Luisa Ferreira. Sorry if I mispronounced names, and my apologies. And thank you for Fernando for coordinating and initiating this event. It's definitely a great opportunity for the two labs to interact. Just to give you a very brief overview of the Brain Dynamics Center, we are based in Sydney, Australia. We are located in the Westmead Institute for Medical Research, which is a research institute affiliated with the University of Sydney. Although we are located in the Westmead Institute, we've got colleagues who work across a different number of different entities here in Sydney. One, the University of Sydney, the University of New South Wales, the Western Sydney Local Health District, which is the major government state health district over here, and of course the Westmead Institute for Medical Research. And this is, we would have loved to welcome you here in person, but we can't. So in this virtual world, photos is all I have to show you where we are located. This is our building, Westmead Institute, and we are located somewhere around here. Right now we are on this floor and speaking to you. The goal, or the main essence of the work we do at the Brain Dynamics Center is truly towards the better understanding of across a number of different mental health disorders and their treatments. And the primary aim really is to use neuroscience in a way to personalize how we treat different disorders. So in terms of identifying precision markers, developing novel treatments which are more personalized to an individual rather than to a group of to a disorder. So we are treating the individual, not the disorder. And here are some different groups which we have integrated at the Brain Dynamics Center, and we do a number of different studies across these different clinical groups. So we look across different mood disorder groups, schizophrenia, anxiety groups, neurodevelopmental groups, studies in ADHD, and more recently eating disorders. Also, our core knowledge of these disorders is towards how to understand how the brain develops in a healthy fashion. So we have a core interest of understanding just healthy neurodevelopment as well. In terms of measures, our approach is truly integrative and standardized. By integrative, I mean we go across a different number of types of measures. So things like what a clinician would typically assess the disorder, so clinical assessments, questionnaires about how the person is feeling, so self-report type measures, cognitive measures. We've got genetics. We do EEGs, ERPs, autonomic responses. And a component of our group is, interest of a group is towards MRI techniques. And the goal of integrating is because each one of them is a small piece of the puzzle. And by integrating these techniques, truly we want to unravel the mysteries underlying mental disorders. And by standardized, I mean every individual who comes up to any study which is done at the Brain Dynamics Center basically goes to a standard assessment of clinical assessments, questionnaires, cognition. Even the fMRI protocol, EEG protocol is very standardized across these different studies. With that, I'll stop and hope this gives you a bit of overview, and you'll hear more about the work we do to our PhD students who are presenting. And I really look forward to hearing from the students from University of Porto as well. Thank you.
Thank you so much. That was an excellent overview. We didn't really prepare anything quite as well prepared as you. So our group is a bit smaller. Our main focus is perhaps more methodological. So we were very strong on EEG and ERP measures, although we do have work moving into neuroimaging. But not having a scanner just around the corner makes it a bit more difficult. So it's always work in collaboration, but it's of course very interesting work to accomplish. Okay, I think we're roughly on time, but to avoid any deviations, I would perhaps then move on to the talks, if that's okay. Okay, so we're going to have a set of 3 talks in the first block by Rita Barreiros, Brun Mazer, and Elizabeth Harris. Then we'll have some time for questions, perhaps, if there are no big delays, or we can have a little break. This would be the coffee break, but we can only offer the break. There's no way of getting coffee across. It's very hard. And then we'll have our second session, and perhaps I can introduce there. Okay, so then I would invite to the stage Rita, who will be talking about the differential neural circuitry in treatment-resistant depression and treatment-sensitive depression.
Thank you, Fernando. Okay, you can see my screen. This will all work. Hi, everyone, once again. Okay. Yeah, I have the pleasure to be the first one presenting my PhD work. As the title suggests, I'm focused on studying treatment-resistant depression. And to give you a bit of an overview of what my project consists of, I will show you, some of you have seen it already, I'll show you a 60-second video that I prepared for the University of Sydney that summarizes the main idea of my project. Hopefully you can hear it as well. Let's see. No. No. Oh.
First, it proposed to dysfunctional connections in our systems to the brain networks in places of darkest brains. Then, we will try to regulate these networks using neurofeedback. Patients rely on MRI scans while repeated use of spectrum the brain is trained, the more that connection is built. It's like a great coincidence. What we will contribute to today is this first part.
Okay, I imagine you got the main idea. So yes, basically we are preparing a study on the efficacy of a neurofeedback intervention for patients with depression. But before I go into the neurofeedback part of it, the first part of my project is to use functional connectivity to study the neural networks, the neural architecture of patients with treatment-resistant depression and patients with treatment-responsive depression. And we aim to use functional connectivity using fMRI data to do this, and I will explain the methods in more detail later. And we are going to look at these through different lenses, but the very first study, the one I'm presenting to you today, is focused on the habenula. The habenula is a small midbrain structure that is known to be associated with anhedonia. Anhedonia is the inability to feel pleasure, and it's a very common symptom of treatment-resistant depression, and this structure is shown to be disrupted in these patients in particular. However, what is not known is how does this structure communicate with the rest of the brain? How is it connected to the rest of the brain? And this is what we are going to try to explore, or we are exploring. For this study, we collected data from 40 patients with treatment-resistant depression, which I will refer to from now on as TRD patients, 40 patients with treatment-responsive depression, or patients who recovered from their depression with medication. We are going to refer to them as TSD, and 40 healthy controls. We defined treatment-resistant depression as symptom remission after at least 2 sets of different trials of antidepressant medication for at least 6 weeks in an adequate dose and frequency, and the presence of moderate to severe depressive symptomatology measured by a score of 16 or more in the Hamilton Depression Rating Scale. And the treatment-sensitive or responsive depression patients were characterized by symptom remission, so they recovered from their depression, they got better to a point in which they rated 9 or less in the Hamilton Depression Rating Scale, and they had antidepressant treatments in the past. All the participants underwent, the whole protocol that Mayuresh described before, the integrative protocol, but here we are focusing on the MRI data. They laid in the scanner for 8 minutes doing a resting state task, and in a 3 Tesla Siemens scanner, and they underwent a comprehensive clinical interview as well. This is a table that summarizes the clinical and demographic information about this data set. All patients were comparable for age and gender and education level. And as expected, there were differences between the groups in terms of current symptomatology. But interestingly, as you can see in the green bit, for the same number of previous episodes, so for a similar chronicity of their condition, the treatment-resistant depression group had a higher level of hospitalizations in the past, ECT treatments, suicidal attempts, and higher severity in their worst major depressive episodes. So we seem to have a more severe cohort in general for this patient population. We had to exclude 4 participants for ineligibilities and for excessive head motion during the scans. All the functional MRI images were processed. We used the standard Korn toolbox pipeline for this. The images were realigned, motion corrected, co-registered to the anatomical T1 images, segmented, smoothed, denoised, and normalized to the MNI space. And for the analysis, for the functional connectivity analysis, we used an approach called seed to voxel analysis. And what does this mean? This means that we selected a priori a region of interest that we wanted to see how this region connects or communicates with the rest of the brain, with each voxel or element in the brain, in the whole brain. So we calculated the functional connectivity measures between the left and the right habenula. We chose these ones as our seeds of interest for this study, and we calculated the connectivity between these regions and each voxel of the brain. And then whole brain-wise, we compared between the 3 different groups using an ANOVA. And we identified particular clusters in the brain that were differently connected with the habenula between the three groups. Then we extracted beta connectivity values from each voxel of these particular clusters for each group, and we compared them as post-hoc tests. To identify the habenula, there was a bit of a challenge, and we used an approach used by other authors in which we visually identified and segmented the habenula using the anatomical images for each patient. We identified the central coordinate for this left and right habenula. We created a 3 millimeter sphere around each of these coordinates, and we created our seeds this way, manually for each participant. This is a table that summarizes the main results at a whole brain level. So how is the habenula connected or differently connected with the rest of the brain when we compare between the three groups? So the main result was the connection between the left habenula and three main clusters in the brain, the precentral gyrus right and the bilateral precuneus cortex, which is part of the default mode network. Then, as I explained before, we compared between the three groups for the voxels within this most significant cluster, and we found differences between each pair of groups with a really interesting pattern of hyperconnectivity of the habenula in the treatment-resistant depression group as compared to the other two groups. And in the left-hand side, you see an image in which you can visualize and locate where these clusters are in the brain. We also ran some analysis trying to look for correlations and associations between this neural measure and this neural finding and the clinical variables that you collected. And we found an interesting correlation between, or differences, we found differences between the disconnectivity of the habenula with the precuneus cortex in treatment-resistant depression patients who had and didn't have a history of suicidal ideation. What does this all mean? You might be asking. I ask myself that a lot too. But from what we've been discussing and reading about this, we know that the habenula is highly involved in the influx of negative thoughts and information, and then it's feedback into the reward system. And we believe that its hyperconnectivity in these patients might be underlying the exacerbated anhedonia in these patients. So in other words, it seems that treatment-resistant depression patients are overly processing negative stimuli. And we also saw that there are some abnormalities in the interaction between the reward system, which is the habenula in this case, and the default mode network structures, in this case the precuneus cortex. So these abnormalities between these two networks might be related or associated with the finding on suicidal ideation, and might be underlying the tendency of these patients for higher history of suicide. There are some limitations in this study that we can acknowledge. The habenula volume is very small. This is a very tiny structure. So we believe that we might be getting signals from adjacent structures, which might be a challenge in the identification of pure habenula signal, and might be interfering with our results. The sample size of the population is pretty small, we know that. And we would also like to see the effects of different antidepressant treatments in these patients. So we collected data from patients who were resistant to antidepressant medication in general. We didn't specify which ones. So this could bring interesting insights as well if we had access to that, maybe in the future. And we are planning other analyses in terms of functional connectivity using other regions of interest, like regions of the default mode network. And we are also planning data-driven approaches like ICA decomposition and analysis. In a nutshell, we thought this is a pretty important finding because it might be giving us a signature marker for treatment-resistant depression. And it is very important clinically to identify in the brain of the patients differences between different types of depression so clinicians can use these techniques to identify them and start appropriate treatment earlier. This is the dream. If you want to read more about this study, it was recently published on Neuroimage Clinical, so it's already available online, and it was presented as well in a couple of conferences. Thank you very much for listening. This is the team at the Brain Dynamics Center. Thank you all. And this is a nice picture that I actually found today. This is Rita, a 20-year-old Rita, and the lab in Porto being a guinea pig for the installation of the EGI system when you just got it, which is funny because we just had ours this week, yesterday. So we have a similar picture of Mayuresh using them. Thank you all.
Thank you so much, Rita, and an excellent talk and an excellent keeping of time. So we would leave questions for the end of the three talks, and so I would now pass on to Prun, who will be talking about a neurophysiological approach to abnormal predictive processing in autism and schizophrenia.
Yes, let me try to share my screen. I have two screens, so I hope I share the right one. Okay. Not anything for now. Is it the presenter view or the right view?
It's
the
good,
presentation?
Yeah.
Yeah, okay, good. Hi, so my name is Prun. I'm a PhD student in the lab that I am just starting. So I'm going to give an approach to the project that I am just starting. And I wanted to take this opportunity to have some input from a lab and a group that has been studying mental illness and specifically schizophrenia. And I thought this would be a good opportunity because I can still change a few things on my experimental design and the whole design. And so humbly, I hope that you would give me some tips about the study in the field of mental illness. So I won't have any data yet, but just the idea of the project, I think it would be interesting. And hope not to disappoint it. So I'll start with talking about the predictive processing framework. This framework theorized that the brain activity develops an explanatory model of the world based on prior experience instead of being simply a passive receiver of information. This model is used to anticipate inputs and help us to adjust to a world in a more efficient way. And so if the model is accurate, then it correctly predicts sensory inputs. Sensory inputs that could come from ears, eyes, but also interoceptive inputs such as temperature, heart rate. And if the model is not correct, then a prediction error occurs. And this prediction error will then be sent back to higher orders of the brain to update our models. In that sense, prediction error would be a mismatch between brain's top-down prediction and bottom-up sensory inputs. And this is an important line to keep in mind for the rest of the presentation. Our goal would be to minimize prediction error, even if it does not describe the real world. We don't need the real world. We need a world that makes sense, that we can adjust to. To minimize prediction error, we would have two ways. We can update our expectations by making better predictions, or we can adjust our sensations by positioning our sensory organs in a different way to receive different sensory samples that can better fit our predictions. And so only the prediction error is sent to higher orders of the brain, like I said before. But not always our sensory data is reliable. So sometimes it can be noisy. And so when sensory data is noisy, we will put more weight on our prior predictions and less weight on our sensory data. So we don't have a prediction error that is wrongly attributed. When our sensory data is more trusted, then we'll put more weight on our sensory data and less weight on our on our priors, which is the image on the left. It's trying to very well define. And so just to give you an example, in a foggy day, we will trust less our eyes because we don't see that well, and we'll trust more on our predictions, for example, that our car is coming. But on a sunny day, then we can trust our sensory data better. And so how does this fit in schizophrenia? In autism first. So the heterogeneity of both actually autism and schizophrenia is puzzling, and it involves several areas of the brain and several processes, but somehow they all fit on the predictive processing framework. In autism, the abnormalities in predictive processing state there is an attenuation of predictions named hyperpriors. So here, when you see the minus, I can point that, we would say these are down, so hypopriors. And autistic individual might put less weight on their prediction and rely more on inputs, leading to a greater prediction error. So more on our sensory organs, and this will cause a greater prediction error. This could explain symptoms such as difficulty to adjust to unexpected events, preference for routines, hypersensitive to noises, but also fits in the reports of superior performance, such as less susceptibility to illusions or enhanced visual tasks, where the lack of priors could actually help in these tasks. Some authors also consider that only the fact that the sensory data is weighted more highly leads to an inefficacy in updating the prediction model. So even if we don't have hyperpriors, which is very theoretical yet, only the fact that we put more weight on our sensory data might lead to this greater prediction error. And so considering this hypothesis, we can actually infer that autistic individuals see the world as more real than non-autistic because they are not as influenced by our predictions and our beliefs. Well, and schizophrenia appears to show the opposite pattern in the prediction processing framework. The sensory signal is imprecise and less trusted, leading to a strong reliance on top-down precision. So the sensory signal is less trustable when it should be trusted, because this could also happen in a normal pattern. The reduced weight on this environmental information and the over-reliance on one's own predictions might explain delusions and false beliefs. And this model starts with the false prediction errors that propagate further in higher order areas of the brain and eventually lead to a not real explanation of the world. Regarding hallucinations, which is the best symptom that we can explain, hallucinations are the excessive sensory activation without an input, which is consistent with more reliance on one's prediction and less and not trusting our sensory information. But how can we test this? How can we measure prediction error? Well, we know that the amplitude of electrophysiological responses, ERPs, is enhanced by unexpected events, even at early stages of processing. For example, if you think of a simple oddball paradigm, deviance or less frequent stimuli elicit a higher response. So in that sense, we can consider that this higher response to unexpected events is a prediction error. And I would include two of the most studied ERPs, mismatch negativity and P300, which are very responsive to simple oddball tasks. And so if these are so well studied, we should know something about these ERPs in schizophrenia and autism. And what we know about a few meta-analyses, actually a lot in schizophrenia, less in autism, we would expect that these ERPs would represent prediction error. And as you remember, in autism, prediction error would be augmented, enhanced, and in schizophrenia would be less, would be down. So what we see is a robust and frequently replicated finding in diminished amplitudes in mismatch negativity and P300 in schizophrenia. However, results in autism are not so well defined, mainly because there's some discrepancies found between children and adults. A meta-analysis by Schwartz and colleagues in 2008 found a tendency for enhanced amplitudes in adults with a small effect, because then we have less studies with adults too. But the opposite was found for children, also with a small effect. And in P300, we have a mix of effects. Sometimes down for children. Actually, this amplitude seems to be diminished in children, but in adults also always reports heterogeneity of signals. Okay, so how are we studying all this in a single project of 4 years? I'm actually at the stage 1 of a systematic review. We wanted to include a continuum of severity from autism to schizophrenia, not only the clinical samples, but from a community point of view too. So we don't know a lot yet about autistic and schizotypical trait scales in healthy adult samples. So trying to have the subclinical groups, high trait groups. And so now we are starting with the scoping review and method analysis with my colleagues that are seeing this presentation. We are preparing this manuscript and this in research, first in autistic traits, because it's where we find the most heterogeneous findings, and we will hopefully do the same for schizotypal traits. And after that, hoping next year, November, October, November, we'll start with stage 2, which is our first experimental study with a large community sample, a non-clinical community sample, and we will use 2 self-report questionnaires, autistic spectrum quotient and schizotypal personality questionnaire, to measure traits in this sample, and then we'll perform 2 tasks that I will show later. And in a later stage of the project, we will do a clinical experimental study with 2 clinical groups of autism and schizophrenia, and we'll have a control group that we should have from the previous study. This previous study with the community sample will also help us to adjust our tasks to the clinical participants, which will need, which might help to adjust with the time that they will spend in the lab, and it's more critical. So in a very simple way, these are the tasks that we design, very simple classical oddball tasks with frequent stimuli as standards and infrequent stimuli as targets. We included visual and auditory tasks because in schizophrenia, most symptoms such as hallucinations tend to be auditory sensations. And on the other hand, in autism, there is a tendency for visual abnormalities. But as I said before, we will see in the community sample if the 2 tasks are needed, if one of them is showing more results, and if we can take out one, maybe for the clinical part. We will also have a noise manipulation sensory information. So we'll have a block of no noise condition and 1 block with noise. And this noise will create a sensory data that is less reliable. And we will hopefully see that prediction error might be diminished if the sensory data is less reliable. Of course, this would be in controls. We will see what would happen in autism and schizophrenia. So what we expect would be this would be the perfect results for us that would match our hypothesis. So in the noise condition, we will have an enhanced amplitudes representing prediction error in schizophrenia and autism. And in the no-noise condition, this would be even higher. We might see some difference in the noise and non-noise condition for controls. And in terms of the community samples, we will hope for a positive correlation for autistic traits and a negative correlation for schizotypal scores. So higher amplitudes for mismatch negativity and P300 and in autism, and the opposite pattern in schizophrenia. And so with this, okay. So we aim to understand the mechanisms that underlie schizotypal and autistic traits in clinical and subclinical populations, considering the predictive processing framework, and also to contribute to improve the knowledge on the search for neurobiomarkers in autism, spectrum disorder, and schizophrenia. And I will just leave the reference. I didn't put a nice photo. I forgot that. But I think I kept the time. Thank you.
You did indeed. Everyone's time tracking is excellent. And so now for the final, the grand finale of this first block, I would move on to Elizabeth Harris to talk a bit about the neural connectivity of the subnuclei of the amygdala in PTSD.
Okay. So hopefully you can see that. All right. So thank you for that introduction. Yeah, grand finale, no pressure. So I am another PhD student in the Brain Dynamics Centre here. I am from the University of New South Wales, so different to most of the people here who are at University of Sydney. So I'm actually working with the Traumatic Stress Clinic, I suppose, department there. So my interests are more in the PTSD area. So I'll be telling you a little bit about one of my PhD projects looking at the structural covariance of amygdala subnuclei in PTSD. So a little bit about PTSD first. So about 60 to 70% of Australians will experience a traumatic event in their lifetime. And of these, about 20% will go on to develop PTSD. PTSD is characterised by persistent re-experiencing of the traumatic event, by avoidance of trauma-related stimuli, by a pervasive tendency for negative feelings and thoughts, and by an overactive reactivity and arousal system. Now, over the past decade or so, neuropsychiatric disorders have been increasingly considered as disorders of brain circuitry. And what this means for PTSD is that brain circuits involved, or brain circuits termed the default mode network, the cognitive control network, and the salience network are all thought to be impaired. And of particular interest to me and to us here is a structure that's involved in the salience network, the amygdala, which is consistently found to be hyperactive in PTSD. Now, I'm sure that we're all familiar with the amygdala, and we've all come across it at some point in our day-to-day lives. The amygdala is a small structure deep in the brain, and exaggerated activity in the amygdala is often seen in response to trauma-related stimuli in PTSD. So it's thought that dysregulation of the amygdala is a key component in PTSD. the amygdala is usually imaged as a single structure, but we can see from the image here that it's actually made up of a number of substructures or subnuclei. Now, a lot of these subnuclei are very small, so they're often grouped together based on similar processes. And we have the three main groupings on the right here. The centromedial nucleus, which is the smallest of the three nuclei, but it's the main output of the amygdala. The superficial nucleus, which is part of olfactory processing, and the basolateral nucleus, which is the largest of the three, and it's heavily involved in associative learning and in processing of emotional stimuli. Now, most of the neuroimaging of the amygdala subnuclei uses functional MRI and functional connectivity, which, for those of you who aren't familiar, is when two spatially distinct areas of the brain show similar patterns of activity across the same time span. But the problem with this is, as you can see in the picture, it's not quite clear. It's heavily pixelated. And when we're looking at the connections of such small areas, as Rita mentioned before, these areas might overlap and we might not get accurate results, which is a problem. So we thought we would use structural MRI instead, which does actually give us a clearer picture. And we would look at the connectivity of the amygdala in this way. And one way to harness the power of structural MRI is to look at the grey matter volume of different brain regions. So at a population level, individual differences in one area of the brain often co-vary with individual differences in another region of the brain. So, for example, grey matter volume or greater grey matter volume in the hippocampus often co-varies with greater grey matter volume in the amygdala, and at the same time with lower grey matter volume in the caudate nucleus. So this process is thought to reflect developmental coordination or even synchronized maturation. But what's important to note is that these regions don't have to be anatomically connected. It's actually their involvement in the same processes that drives their connection. And in this way, it's thought to be related to functional connectivity. So just to recap before I go on, PTSD is increasingly being considered a disorder of the brain circuits, and it's consistently found to involve a dysregulated amygdala. The amygdala is a structure that's made up of many subnuclei, but these connections are really difficult to see with traditional fMRI. So we thought we would use structural MRI, and particularly structural covariance of grey matter volume of brain regions, to look at these connections in both a healthy control versus a PTSD group, but also within groups. So data for this project was actually collected as part of a larger project at the Brain Dynamics Centre, and we used the 3T-T1 weighted structural MRI scans of 67 PTSD participants and 71 healthy controls. And in terms of the pre-processing, what we did was we obtained the grey matter volume of the 3 regions of the amygdala subnuclei for each hemisphere. And we got the correlations between these 3 regions and 92 other regions of the brain. And this essentially gave us 2 correlation matrices for the healthy control group and for the PTSD group, which were the basis of our analyses. And as I mentioned before, the analyses consisted of a comparison of these correlations between groups and a comparison of the different subnuclei connections with the other brain regions within each group. Now, unfortunately, we didn't actually find any group differences in the structural covariance of the subnuclei, nor did we really find any differences in the healthy control group. However, within the PTSD group, we did find some differences. So what we can see here is the brain on the left is the difference in the superficial nuclei versus the centromedial nuclei. And what we can see is that the right cuneus actually showed more or higher covariance with the right superficial nucleus than it did with the right centromedial nucleus. And that's the only result we can see that's unique for the superficial versus centromedial nucleus. On the right, however, we can see that the bilateral basolateral nuclei actually covaried with a number of regions more than the centromedial nuclei did. And we can see these regions are centered around the temporal lobe and the medial and lateral frontal lobe. And what we can also see, and what I've circled in different colors, is that the regions that the basolateral nucleus is covarying with are regions that are actually associated with the reward, the attention, and cognitive control networks. And if you'll recall, these are networks that we do see that are impaired in PTSD. So if we look at these results collectively and try and ground them in the context of PTSD, they do seem to suggest a few things, and we are still working on these interpretations, so I'm happy to hear anyone's thoughts on these. So number one, the connection between the basolateral nucleus or nuclei and areas involved in somatosensory and social interpretation. These areas could potentially underlie a heightened attention network that basically primes the system to be more attentive to negative related information at the expense of positive information and positive affect. What the basolateral amygdala connection with the precentral gyrus and ACC could suggest is something to do with the cognitive and inhibitory control of behaviour, as we see that these connections are actually found in other human studies and in rodent studies as well. And these studies in humans and rodents were looking at the cognitive and inhibitory control of behaviour. So these areas are impaired in PTSD too. And finally, and complementary to this result, the connection or covariance between the superficial nucleus and the cuneus could underlie a compensatory mechanism for this dysfunctional frontal inhibition that we see with the BLA. And this interpretation is based on a study that shows that higher cuneus activity is actually seen in PTSD in relation to trauma-related stimuli. So these are the three kinds of main interpretations that seem to fit together with the results. So just before I finish, I want to leave you with a few takeaways from the presentation today. So number one is that structural covariance is a really good tool for looking at small regions of the brain and for getting a clearer picture of these small regions and their connections. And because the results seem to marry up quite well with functional connectivity, it's a good adjunct to functional connectivity analyses. Number two is that the amygdala as a whole is important to PTSD, but so are its subnuclei. And I think it's important that we start looking at the distinct contributions and connections of these subnuclei to see how they differentially contribute to PTSD and other neuropsychiatric disorders for that matter. And number three is that attention and cognitive control networks are important in PTSD, But more and more, we're seeing that areas involved in the positive affect and reward network, as Rita also found in depression, are impaired in neuropsychiatric disorders and in PTSD. And so we should be looking at these sorts of networks as well when we are actually investigating the brain regions involved. So yes, I don't have a pretty picture. I wasn't at that conference, but I would like to thank my colleagues here and at UNSW Psychology, and thank you all for listening.
On the first half, thank you to all the speakers. And now we have a chance. Let me sort this. Okay, now we have our time for questions, and for if anyone really needs a break, now is the time for that. But I would suggest we take the time to just be able to talk a little bit. If there are any questions for the speakers.
Yeah, I have a question.
Gustavo, is that, yes?
Yeah. So I have a question for Rita.
Yeah.
And actually, this is a coincidence, but just yesterday I was looking into treatment-resistant depression, and I was looking into deep brain stimulation possibilities, so different areas that could be used. Apparently, there haven't been a lot of good studies. Some of them are just case studies. Some of them are just with mice. So one of the regions that they mentioned is the abinula, precisely. So I thought, hey, I should probably ask you, do you think that it would fix just a section of the symptoms, which then would facilitate the rest of the treatment, so to say? Or do you think that it will probably affect all of the symptoms that are related with depression? Because different areas could affect different symptoms, I assume.
Yeah, so you're wondering if modulating directly the functioning of the abinula would have an impact in the one symptom I mentioned, for instance, the anhedonia, or the whole set of symptoms in depression. I don't know the answer to that. I guess that's the answer to that could be in studies that look at the efficacy of different treatments that target the abinula, and the project that we want to run with neurofeedback, for example, would be one example of that, in which we are going to target the function of particular regions of interest in the brain or connections in the brain. Can be the abinula, can be others, and we are still going to see if it is possible to modulate that to start with, and then we will see what sort of clinical outcomes we get from that intervention. So I guess it would change case by case. I saw some studies with using ECT treatments and TMS treatments targeting the abinula in treatment-resistant depression, but I never saw something like one symptom being changed and the others don't. I never saw such thing. I'm not sure if Anthony, so Professor Anthony Harris is, hi Anthony, is a psychiatrist, and he's a co-director of the Brain Dynamics Center too. I'm not sure if you want to add anything onto this comment. You're on mute.
Yeah, I'm off mute now. Look, I think you're quite right that there's no specific area for depression. It reflects dysfunction in many different parts of some underlying neurocircuitry. So I don't think that just hitting the abinula is likely to be the answer to all treatment-resistant depression, if it ever was a single entity. But it might be part of the answer, and one area which might be reasonable for specific patients.
Thanks, and thanks, Gustavo, for the question.
That's it, thank you.
Anthony, you have a question?
Yes. I had a question for. We're just about to start off on an electrophysiological investigation of auditory hallucinations in people with schizophrenia, and a number of other different disorders. And so I was interested in the comparison that you're making with autism. So a couple of questions. So one is that when you say the community sample of people who have both sort of autistic and you'll be able to look at a tray of autism and a tray of schizotypy, there's of course going to be overlaps, quite significant overlaps. So I suspect is your community sample that well-known sample called psychology students? So I was just wondering.
Hope not only psychology students.
Hopefully not only psychology students.
I'm not a psychology student, actually. I'm a neurophysiology. I studied neurophysiology before, so I'm a bit of an outlier in the lab.
Excellent. Well, I'm not a psychologist either, so I was just wondering how you're going to measure that. And my understanding is there will be a significant overlap in a group of the population. And so that might give you an excellent chance to look at, to compare the effects of the two different trays in your populations. I was wondering what your prediction would be for that group which will have both autistic and schizotypal trays.
So I would hope that the population that would have high traits of autism would have the opposite pattern for schizotypy. But
if they have both those trays, and I'm sure you will find a group which have got both trays. So I mean, I think it's an interesting thing for you to think about. The other thing is whether when you're talking about high and low effects for the P300, I assume you're talking about amplitude.
Yeah.
So there is quite a variation in the latency of the waveforms of the P300, and that was thought at one stage to be part of the reason why P300 was reduced. And so I think that might be an interesting thing to also look at in your meta-analysis, the latency of the waveforms in autism, which I don't have a clue. And finally, just what are you going to do about medication? So a lot of the people with schizophrenia will be on antipsychotics, and that's unlikely. You'll have some people with autism on medication, but probably far fewer. And I think that's an issue. So I was wondering what you'd be doing with that.
Yeah,
these are very difficult questions. I don't know anybody who can satisfactorily answer the last one.
Yeah, thank you. This is great. So probably I will use the medication as a covariate. I have one of my co-supervisors is a psychiatrist, a medical doctor, and I will seek her advice too in that matter. She will be the one responsible for helping with the clinical groups. But I probably would use, as I've been seeing in most of studies, use it as a covariate that we have no chance to say, don't take the medication this week. So I'll have to adjust. But yeah, a lot of problems will occur probably. But I was thinking still about the high traits of schizotypy and autism. And well, I have to see what we get, but maybe a group that has both high traits in both would be also.
I suspect you'll find a group.
Yeah,
high traits in both.
Too interesting to see the results in that group specifically. Maybe that would be nice. Thank you.
Right, everyone, make sure you take notes. Okay.
I'm taking.
We'll be talking about this in our next meeting.
Yes.
Mayuresh?
Thanks. That was a really good talk, Poon. You really explained well the PhD project, and it's a very fascinating project. Just adding to that, the discussion which was happening, and just my small contribution, because you're looking at healthy individuals, I wonder if you would consider doing like a clustering analysis so that you actually pull across the two cohorts and cluster them based on either only schizotypy, only autistic, and a group of overlap. And if that would give you some sensitivity in looking at differences in the measures that you're interested in.
Yeah, I think that's exactly what I will do. I think that's very nice. That was a statement, right? Was not a question. Was you have
just
giving me? Yeah, thank you. That's great. I will do that.
I just raised my hand because I also have a question rather than just moderating for Elizabeth, actually. I thought this approach of using structural covariance was really interesting. Just a couple of related questions. The first one is there evidence that this relates to functional connectivity? So have there been validation studies, or this is more of something you would expect to see?
I don't believe there have been validation studies, but people have looked, the results, as I mentioned, that people do get from structural covariance studies and functional connectivity do kind of show the same thing. So, yes, I don't.
Alexander Block has done the closest thing to validation studies. Yes. And they've given values of the correlation between the two modalities. Yeah. And off the top of my head, I can't quite remember that. I thought it was around 0.6 or so for DTI and for DTI, sorry, and structural. Functional, yeah.
So, I mean, just because of the way the technique works, it's rather than diffusion tensor imaging or diffusion weighted imaging, which is more hardwiring, structural connectivity overlaps more with functional connectivity. And like Christy mentioned, there's some work validating that overlap.
Well, I mean, it might be an opportunity for a methods paper somewhere in there to kind of show that that you can get reasonably similar measures. Okay, so we have a comment in chat. Carlos, you want to speak up or?
Yeah. Hello, everybody. I just didn't know about the time, about the timing. So it was regarding like the discussion on the medication of schizophrenia patients. I know that you can use like the chlorpromazine equivalent dosage that you can like compute for each type of antipsychotic, like an equivalent dosage of chlorpromazine, and this may be an approach for controlling like the medication in schizophrenia patients to see whether it correlates with the experimental modulations that you are trying to do. Although this will not account for the different mechanisms that each antipsychotic may induce in the patients. And there's the problem with like the interaction of medications, because from my experience, I worked with schizophrenia patients before, like the average number of medications is about like 2.5 or 2 point something, because some of them are also taking other medications. Yeah, this is challenging, but this is the only way to do it, right? We have to try to control this somehow. But great talk, thank you so much.
You know that when you will be in the lab, I will be talking to you all the time. So how did you do with your patient? in a year.
Perfect.
Yeah, this is well kind of the standard way of correcting for this statistically, but it doesn't solve the problem. It just kind of gives us a notion of how big the problem might be. I would argue.
Yeah, that's exactly it. Yeah.
Okay, we still have a couple of minutes. Okay. Well, I guess we've run out of questions for now, and so perhaps we could move on to the second block of talks. And so I would now call upon Katerina to come talk to us about exploring psychopathic traits in adolescence.
Okay, let me just try and share my screen. Okay, is everybody seeing this?
Yes.
I am trying to put it in presentation mode. Okay, so hello, good morning, and good afternoon, everyone. My name is Catarina Prata. I am a PhD student at the Laboratory of Neuropsychophysiology, and I am going to present to you my still very ongoing PhD project, which is called Exploring Psychopathic Traits in Adolescence, Behavioral and Neural Dissociation of Grandiose Manipulative, Impulsive Irresponsible, and Callous Unemotional Traits. So, of course, the main concept of my project is psychopathy. And historically, psychopathy has been approached through taxonomical approaches and as a unitary construct. However, more recently, we have dimensional approaches, and one of the most known by now is the triarchic model of psychopathy that was developed by Patrick and colleagues, and that one is fundamental to my project. So this model integrates 3 different phenotypes, as you can see here: disinhibition, meanness, and boldness, and diminishes the focus on antisocial behavior and considers psychopathy like a dimensional construct, so with traits distributed in a continuum. And even though these 3 constructs interrelate at some levels, they have different phenotypical identities. So, for instance, disinhibition means a general propensity for impulsive problems, and it's not specific for psychopathy, and is considered, of course, maladaptive. And similarly, meanness includes empathy deficits, cruelty, and others being considered maladaptive as well. But boldness, however, includes the capacity to remain calm in stressful situations, being able to controlling themselves. So it's considered in itself an adaptive trait. And it's what differs psychopathy from antisocial behavior. So a phenotype is not by itself the concept of psychopathy, and so the study of their dissociation is needed to understand the phenotypes better. And recently, as you can see, there's something here, because the studies have been directed to younger samples, since psychopathic traits can be gauged consistently since school age with a trifactor approach based on the adult original one. However, the studies focus more on the callous and emotional traits, so the meanness part for young adults, and less in others. So it is important to focus on the sociable dimensions between these traits to understand different dimensions and intervene specifically. So the goals or objectives for my project are to analyze psychopathy in adolescents, to study the dissociable effects of the youth's psychopathy traits, to contribute to a dimensional approach to psychopathy, and to conduct an exploratory study focused on the mother-youngster relationship. And you may ask, okay, but where does this study fall in here? Studies show that parental psychopathy traits may influence not only children's psychopathy traits, but also psychopathological results. So that said, it is necessary to study not only youngsters, but their parents as well. So talking about this, my sample is going to be 77 youngsters, ranging from 12 to 16 years old, and their respective mothers, and they'll all be recruited from the community. They'll be free of neurological, psychiatrical, and motor disorders that would impede them to do the EEG tasks. And for the youngsters, I will do some questionnaires like the Youth Psychopathy Inventory, the Basic Empathy Scale, the State Trait Inventory for Cognitive and Somatic Anxiety for Children, and the Impulsive Premeditated Aggression Scale. For the mothers, I will only focus on the triarchic psychopathy measure, since I want to understand the relationship between the psychopathic traits in youngsters and the mothers. And so the EEG will be a 128 channel system. So I'm going to have 3 different tasks for them to do, since each of the tasks will be directed to one of the main groups of the psychopathic traits in the youngsters. So I'm going to have an aversive cognitive task, and that task is the one here up above. And that task has 3 phases: a phase of habituation, where they only see gray circles and nothing else; then a phase of acquisition, where they start seeing coloring circles, and for one specific color for the participants, half of the circles, more or less, will have a very high-pitched static noise that is aversive, of course, and half of them will not. So there will be paired and unpaired stimuli. And then, of course, we will do the extinction of it. So this is important because higher traits of boldness or grandiose manipulative in youngsters normally present less startle, less anxiety, and obviously less aversive conditioning in here. Then we go to a go-no-go task where we have two very similar images with some slight differences. And in here we have a ball and a clock. So the meaning of this task is to tell them, okay, so you'll have to press a button when, let's say, for instance, the ball appears, and you will have to withdraw the pressing when the clock appears, for instance. And the idea is to do this extremely fast so that we can see if there are mistakes made. Of course, if they have higher traits of impulsivity and irresponsibility, they will commit more errors. And so we will want to see if this is true or not. And then we have the last task, which is an empathy-inducing task, and this one is very related to meanness. So we'll have images of hands and feet, and those images have two blocks. So one block will be images that are not specific of pain. They are near something that might cause pain, but they don't have pain specifically. And then we have the other block where already are images that could be associated with pain. And the idea is if they present more callous and emotional traits, we will see less empathy in this task. So for the statistics that I want to do two types of multiple linear regression. So for the first one, the predictors will be the psychopathic traits for the mother and the youngster, and the results will be the behavioral and neurobiological data. And for the second one, the predictors will be the mother's psychopathic traits, neurobiological data, and behavioral data. And the results will be the youngster's psychopathic traits, neurobiological data, and behavioral data. So we can see if there's a relation in here. The road so far has been very contaminated by COVID, as all of us know, I assume. And so I haven't been doing any of this. I'm still recruiting participants, so I haven't been in the lab with them. However, I have searched and done some background studies. The first one is a mini systematic review called Event-Related Potential Correlates of Aversive Conditioning in Psychopathy. And that review has been submitted to Neuroscience Letters, and it's currently under review. I can systematize a bit of the main findings for you, which is self-reported results suggest that higher psychopathic traits overestimate unpleasantness in both paired aversive stimuli and unpaired aversive stimuli. What does this mean? This suggests that higher psychopathic traits and people with higher psychopathic traits fail to discriminate threat-related contingencies associated with both stimuli. Evidence leads to the hypothesis then that psychopathics cognitively understand that the presented stimuli are supposed to be aversive, but emotional deficits may prevent them from devising accurate estimates of effective ratings. For early ERP results overall, they appear to unveil deficits in the neural processing of aversive stimuli in psychopathic participants. The results are in line with the low fear hypothesis. And for later time window ERPs, they further support this hypothesis. Since these components are thought to reflect high-order cognitive processes, such as attention, a smaller amplitude suggests that psychopathic individuals show less attentional allocation for processing threat cues. However, of course, more research is still needed. And the second paper that I'm working on is a meta-analysis called Amplitude Modulation of the Contingent and Negative Variation in Psychopathy, so the CNV, but it's still very ongoing in the beginning, and so I don't have results to show you yet. So that is all. Thank you so much for your attention, and I hope that you enjoyed my presentation. If you have any suggestions or any questions, I'll be open to that in the end of this block of presentations. Thank you so much.
Rita, that was a record time for your talk. Thank you. And so we now move to Sabine Rai. Investigating functional neural circuits in euthemic bipolar and major depressive disorder. Sabina, please go ahead.
Hello, everyone. So I'm a final year PhD student at the Brain Dynamics Center, and I'm in the final stages of writing my PhD thesis. So I'm going to briefly be giving an overview of my thesis before discussing one of my published papers. So a little bit of background on bipolar. So bipolar is the 6th leading cause of disability in Australia and is thought to affect approximately 2.9% of the Australian population aged 16 or over. The World Health Organization has also labeled bipolar as one of the leading global burdens of disease. So one of the aspects of mood disorders is that emotion regulation is impaired, which contributes towards the mood-based and emotional symptoms that are experienced, such as the emotional instability. And one of these strategies is called cognitive reappraisal. Now,
Celine, you're on mute.
Sorry. Sorry, can you hear me? Sorry, my Wi-Fi is a bit funny, sorry.
We can hear you well now. Before it was just cutting off a little bit.
Yeah, is that better?
It is.
Can you hear me?
Yeah.
Wait, can you hear me?
Yes.
Yes, we can.
So specifically, cognitive reappraisal is one of the most adaptive emotional regulation strategies, and it requires the individual to consciously reinterpret a negative emotional scenario as being less negative. And this is particularly impaired in individuals with mood disorders relative to healthy controls. But there's also a lack of cognitive reappraisal imaging research in mood disorders compared to other less adaptive strategies. So this is one of the gaps that my thesis aims to address. Also, both bipolar and MDD, major depressive disorder, manifest homogeneous clinical presentations. So with the depressive polarity, for example, but they're obviously different at a neurobiological level, which then comes to another challenge, is that up to 40% of individuals with bipolar, when they go to a clinician, are initially misdiagnosed as having major depressive disorder, again, as the depressive polarity typically develops first in the trajectory of the bipolar disorder. So it's important for this thesis to identify possible markers, mechanisms in the brain as well that can differentiate bipolar from major depressive disorder. But also it's important to find a trait-related neural mechanism, because a lot of the previous studies, if they have investigated both disorders together in the same cohort, have looked predominantly at symptomatic cohorts. And while it's great to find neural correlates of depressive and manic episodes, it's also important to find trait-related markers that are present even in the state of remission, because it's important for risk diagnosis, like prognosis, intervention. And so this comes to the overall aims of my thesis, which is firstly, to what extent are emotion regulation neural circuits impaired in bipolar and major depressive disorder? And can we find a clear distinguishing marker within the cognitive reappraisal neural circuits that can discriminate these two disorders? So this was the first paper of my PhD, and use a task-based cognitive reappraisal paradigm. The second paper of my PhD, which is the one I'll be discussing today, is looking at resting state functional activity. And can we identify abnormalities within the functional architecture of the brain that can distinguish euthymic bipolar from MDD? And thirdly, my third paper of my PhD focuses on to what extent can resting state identify both common and differential neural mechanisms in the euthymic asymptomatic bipolar and MDD cohorts, but also using the symptomatic treatment-resistant cohort that Rita was investigating. And so this is the second paper of my PhD, which was published last year in Translational Psychiatry, and its default mode and frontal parietal network connectivity during rest distinguishes asymptomatic patients with bipolar disorder and major depressive disorder. So firstly, a little bit of background. So previous work from my first paper has identified significantly different task-related activation during the cognitive reappraisal paradigm in bipolar disorder compared to major depressive disorder, and namely in the subgenual cingulate cortex, which is also an important region for this paper. However, when you look at task-based fMRI, there can be certain confounds, such as the individual's the motivation, the fatigue, the time of day, but also you're quantifying neural activity for that specific task in question. But with resting state fMRI, you can examine the brain when it's in a task-free state and it's free of these confounds. And you can also look at the intrinsic core architecture of the brain, which may be responsible as well for this task-related performance, and also the organization of the brain. So the triple network model, salience network, which is involved in attending to the emotionally significant stimuli, the default mode network, which is to do with internalized self-referential processing and social cognition, and the frontal parietal network, which is related to cognitive control, is thought to be impaired across both mood disorders. So say, for example, over-reliance on a particular network, such as a default mode network, may account for emotional instability, whereas under-reliance on a network may account for some of the deficits that are observed in, say, for example, bipolar, where they have disorganized speech or psychomotor deficits. And so also as well with previous literature, which adds to the concept of the neurobiology between bipolar and MDD being different, two studies that I will talk about that I also reference at the end. They both used depressed bipolar and depressed major depressive disorder individuals during resting state. And they found that in the individuals that were depressed with bipolar, they had greater connectivity in the frontal parietal network compared to MDD and controls, whereas with the MDD group, they had greater connectivity with the default mode network compared to the bipolar and the controls. And again, in a different study, depressed bipolar individuals were found to have greater connectivity between the salience network and the anterior insula, which is part of the salience network, and the frontal parietal network compared to the MDD group, which found to have connectivity with the default mode network. So based on this literature, we hypothesized that the bipolar group in this study would be expected to exhibit differences in connectivity in the FPN and also the frontal parietal network and the salience network relative to the MDD group and the controls, whereas the MDD group would be expected to elicit differences in connectivity in the default mode network relative to bipolar and controls. So with the methods, we used individuals, 39 euthymic bipolar, MDD, 40 euthymic bipolar, and 41 healthy controls. And both groups met the diagnostic criteria for bipolar type 1 or major depressive disorder. And they were asymptomatic at the time of testing. So the remission was characterized by at least 14 days of scoring under 7 on the Hamilton Depression Rating Scale. We also used the Young Mania Rating Scale and the Depression, Anxiety, and Stress scales as well. And they underwent a resting state scan. So the data was pre-processed and we used two analyses, region of interest and independent component analyses. So region of interest is what Rita described as a priori. And so we took some of the seeds from the 3 networks. So I'll outline just a couple from each one. So for example, in the default mode network, we took the subgenual cortex and the posterior cingulate cortex, among others. In the salience network, it was the amygdala and the anterior insula and dorsolateral ventral cortices. And so with region of interest analyses, you're looking at the connections just between these particular seeds and not with any other regions of the brain. But with the independent component analysis, this is more whole brain. So when you have an MR signal, you have the true neural signal that's coming from the circuits you're wishing to investigate, but then you also have noise coming from people moving in the scanner and breathing. And so what ICA, independent component analysis, seeks to do is to use an algorithm to go in and separate the true neural signal components from the noisy components across the entire data set. And these are called independent components, and they correspond to different neural networks. So here you can see that 32 independent components were identified across our data set, and they all are matched using a correlational Pearson's R approach to the different resting state neural networks in the brain. And so what we basically did was we looked at all of these particular seed-based and ICA analyses. So what we found from the region of interest was that there was significantly greater connectivity between the subgenual cortex and the right dorsolateral prefrontal cortex during rest in the bipolar group relative to the MDD and controls. So between the default mode network, which is the S subgenual cortex, and the frontal parietal network, which is the dorsolateral prefrontal cortex. And then moving on to the independent component analysis. So again, these are the findings, and I'll outline a couple of the key ones. So again, in the default mode here component, that the bipolar group, they compared to the MDD group, had greater connectivity between the default mode network and regions of the frontal parietal network. Both MDD and bipolar group, they shared a common result of increased connectivity between the salience network and visual regions. And then in bipolar here, they had greater connectivity between the sensory motor network and regions here, such as the intracalcarine cortex and occipital fusiform gyrus, which are related to vision and parietal. And so what we can conclude from this is that our findings were consistent with our hypotheses, as the bipolar group were found to exhibit differences primarily in the sensory and attentional networks relative to the MDD group and controls. So firstly, with the region of interest analysis, with the subgenual cingulate cortex, it's related to internalizing emotive stimuli, and then the dorsolateral prefrontal cortex is related to inhibitory and attentional control. So it may be that this particular connectivity feature is related to some of the experiences during the emotional episodes. So say, for example, the highly awareness of the depressive episode, but then another feature that individuals with bipolar assume to have is that they're unaware that they're in a manic episode. So this could be a possible neural mechanism which is underlying these particular features. Moving on to the independent component analyses, again, the key finding, which is loosely mirrors the region of interest analysis finding, is that the inferior frontal gyrus and the default mode network, where the connectivity was altered more in bipolar compared to the other groups. And this, again, may indicate that there is disruption of the frontal parietal cognitive regions, which is coming from the default mode network regions, and may relate to the difficulty that individuals with bipolar have when it comes to regulating and processing stimuli during both manic and depressive episodes. So the other findings, with the deficits in the sensory motor network in bipolar, more so than MDD, may relate to disorganized speech, possible psychomotor deficits, and some of the other symptoms that are observed during mania, and are shared results of the salience network connectivity with visual regions. It could also be a shared mechanism, as individuals with mood disorders are thought to have deficits when it comes to visual recognition. So when looking at face processing paradigms, and there's more biases, negative biases, etc. So this could be related to that. So some of our limitations is that we did use a medicated sample. So there could have been medication effects. However, we did control for this also doing ANCOVers, where we used the medication as a covariate to see if it had any impact that was significant on the neural findings. And this was not the case. Also, we only use a asymptomatic cohort. So if the mechanism is truly trait related that we find, it could be that we should also try and replicate this in symptomatic bipolar and MDD to see if there are similar neural differences. Also, we didn't follow up on the MDD group to check, as it is sometimes possible that individuals with major depressive disorder can switch over to bipolar. But we did calculate the average duration of the illness for our MDD group, and it was 15 years. So that's considered beyond the average duration that bipolar can manifest following a depressive episode. And so the implications of this is that we found that across both a priori and data-driven whole brain analyses, that there was connectivity between the default mode network and the frontal parietal network that was a key distinguishing feature of bipolar disorder relative to the major depression group. And it may be a unique feature of the illness. It may be a trait mechanism that's not impacted by mood states, as inferior frontal gyrus is also found as a risk factor in those that have the genetic predisposition towards bipolar, but are still typically considered clinically healthy. However, future research is also warranted to determine if this could be a neural signature that can be used to classify bipolar from MDD. And thank you for listening. I would like to acknowledge my supervisor, Mayuresh Korgaonkar, and everyone at the Brain Dynamics Center and Department of Radiology. And yeah, thank you for listening. And these are the
Sabina. And now for our second grand finale, I would move on to Carlos.
Hey, hello everyone. I'm going to share my screen. Are you seeing the slide now?
Yes.
Perfect. So good afternoon or good morning, depending on where you are. My name is Carlos, and I'm a PhD student in neuroscience at the Laboratory of Neuropsychophysiology. And I'm going to present my a part of my PhD project, which aims to fine-tune the understanding of the interplay between psychopathy and empathy. And here I'll be presenting the study protocol of the last stage of my PhD project, which aims to use heartbeat evoked potentials to take a deeper understanding into these psychopathy phenotypes. So regarding, I cannot, yeah, okay. So regarding the background of the hierarchic model of psychopathy, so my colleague Katerina already saved me some time because she presented the model that most of us in our lab are working on when studying psychopathy. So this is a dimensional framework of psychopathy that conceptualizes three major phenotypes of psychopathic personality: boldness, which is more closely linked to the adaptive traits of psychopathy, as well, and you have also meanness and disinhibition, more linked to the non-adaptive manifestations of psychopathy. So one thing that it's important in this model is that boldness and meanness typically stem or are argued to stem from the same etiological pathway, which is trait fearlessness. So the boldness and meanness share some variance, which presumably is linked to this etiological pathway of trait fearlessness. And you also have the externalizing pathway, which underlies the disinhibition phenotype, but also has been linked to the meanness phenotype. So disinhibition and meanness also share some degree of variance. So one interesting topic in this field is to try to understand what drives these two etiological pathways. So my hypothesis was that interoception could play a role, and interoception can be defined as the perception and subjective experience of our internal bodily states. So our brain is continuously generating a model of our inner body, about our heart, our bowels, our lungs, our inner temperature, and this provides an inner landscape of the bodily system. And so, taking a look at interoception literature and at the psychopathy literature, the possible link between these two constructs was based on these neuroanatomical arguments. So the main brain region associated with interoceptive processing has been the insular cortex, which is a structure that is widely activated by several interoceptive stimuli. Also, there is evidence from structural and functional brain imaging studies suggesting that the insula is implicated in psychopathy. Also, some of the proeminent conceptual frameworks of psychopathy also talk about the insula in their models. And here I want to highlight the dual process model that suggests that the trait fearlessness pathway that I previously mentioned is associated with impaired activity on the defensive motivational system with the brain, which includes the insula. So within this model, interoception may be linked to psychopathy through this trait fearlessness etiological pathway. So from a mechanistic standpoint, the association between interoception and psychopathy can be framed and tested with the recent interoceptive predictive processing framework. Thanks to Prune, I can spare some extra minutes as well in this slide because she explained this predictive processing framework very well. So, but this framework can be applied to interoceptive stimuli, as the proposition is that we are continuously generating predictions about our interoceptive states, and then we this information, these predictions is compared with the actual incoming inputs from interoceptive signals that can generate interoceptive prediction errors. And there's been some authors arguing that the insular cortex role on interoceptive processing is actually comparing the interoceptive predictions with the actual interoceptive inputs. One way to test this interoceptive predictive processing framework is using heartbeat evoked potentials, commonly designated as HAPs. So these are brain signals pertaining to the cortical processing of cardiac efferent inputs. So we typically compute this by averaging EEG signals time-locked to ECG events, okay? So these heartbeat evoked potentials typically occur in response to each heartbeat, and they are usually found, and meta-analytical evidence has proven this quite recently, that these HAPs are typically found at frontal central sites between 200 and 400 milliseconds after the R peak of the ECG signal. And some studies, including intracortical recordings, have shown that the insula is one of the sources of these HAPs. HAPs can be argued to index precision-weighted interoceptive prediction errors. As you may recall from the talk of Prune, this precision-weighted interoceptive prediction errors stems from the need to adjust to give more weight to the input or to the prediction. And one interesting manipulation that further reinforces how these HAPs can be used to index the precision rating of interoceptive prediction errors is the attentional modulation, where participants are asked to focus on their heartbeats, and this enhanced allocation of attentional resources to the heartbeat actually increases HAP's amplitude, which likely indicates enhanced weight attributed to these interoceptive prediction errors. So we are designing a study protocol aiming to explore how this attentional modulation of HAPs can be associated with psychopathy phenotypes of the triarchic model. So we are trying to modulate HAPs using an attentional modulation paradigm, and we think that this modulation will be differentially associated with the phenotypes. We also be exploring collecting data for interoceptive accuracy tasks to examine whether the psychopathy phenotypes are also associated differently with the interoceptive accuracy performance. And this is important because interoceptive accuracy can be described within this predictive processing framework as the ability to increase the precision of interoceptive inputs by enhancing attention to these stimuli. So we aim to collect at least 50 subjects, a community sample with no history of psychiatric or neurological disorders, no intake of psychoactive medication, and no sensory or motor deficits that can influence task performance. And we'll be assessing the triarchic model with the triarchic psychopathy measure, a self-report measure. Then we have two tasks to measure interceptive accuracy performance, and then the heartbeat attention task that will modulate interceptive attention in the participants. So the heartbeat tracking task is a classical interception task where participants are required to close their eyes and concentrate on their heartbeats and count them in random time intervals. So at the same time, we are measuring the true heartbeat using ECG, and then we compute a score that correlates, it's not like a correlation, but that contrasts the objective counted heartbeats with the subjective experienced heartbeats. So this task has faced a lot of criticism recently. So this is a open debate in the field. So there are several possible confounders, including the instructions. So we are using revised instructions from a recent researcher that found that these played a major role in the performance. Also, there's some confounding. So some people, instead of answering their heartbeats by estimating the heartbeats they are sensing, they use a time estimation strategy, and others use their previous knowledge about the heartbeats that nowadays is more common due to like smartwatches and stuff like that. So we are trying to control these confounders in our analysis. Then we have the emotion interception task. And in this task, we'll be presenting affective stimuli to the participants of several valence and arousal degrees while recording electrodermal activity. Then participants perform subjective ratings of arousal after looking at the stimuli. And then we'll use the correlation between objective arousal as measured by peak skin conductance responses and subjective arousal. And this correlation for each participant will be an index of emotional interceptive accuracy. So it's more like the effective nature of interceptive information, which is interesting for psychopathy. And finally, the heartbeat attention task that aims to manipulate the heartbeat evoked potentials. Here we'll be recording EEG and ECG, and participants will be asked to complete alternating blocks of 20 seconds where they will focus their attention on either the heartbeat or on the white noise that you'll be presenting using headphones. The white noise will be presented in both interceptive blocks and extroceptive blocks. So that way we can ensure that changes in brain activity are caused by the attention shift and not due to changes in sensory stimulation. We are also planning a slightly adapted version of this task that aims to control the cognitive efforts effects, because focusing on the heartbeat, it's much more demanding than focusing on the white noise. So we are trying to adapt this task so the white noise has some slight bursts of volume increase, and in extroceptive blocks, participants have to count these bursts of volume increase, while in the interception task, they have to count their heartbeats. So the goal is to match the task difficulty between blocks as much as possible, which was not done in the original task. So the challenge in heartbeat evoked potential is data processing. As you may know, working with EEG, there's a lot of typically cardiac field artifact. I did a lab rotation in Munich this year or last year, I don't remember. The pandemic got the shadow kind of mixed up, but I did a lab rotation with a group specialized working with HEPs, and they use this very interesting pipeline where they use current source transformation to reduce the cardiac field artifact, and then they use the massive data-driven method, permutation method, to identify the time windows and the electrodes where the HEPs were actually modulated. So we'll be conducting this implementing this pipeline to identify first the electrodes and time windows where the attentional modulation paradigm actually modulated HAPs. And then we'll extract the significantly modulated electrodes to use this as an outcome for the regression models between HAPs and psychopathy scores. So our hypotheses are that first, we estimate that the intentional modulation of HAPs induced by the heartbeat attention task will be positively associated with the performance on interoceptive accuracy tasks. So this is a link between the behavioral data and the neural manipulation. Secondly, as we previously described in the background, so impaired insular cortex functioning, which may be associated with the trait fearlessness pathway, will lead to a negative association between interoceptive accuracy, the HAP's modulation, and boldness and meanness phenotypes measured with the self-report measure, but this will not be present on the disinhibition, which is associated with the other etiological pathway of psychopathy, which is externalizing vulnerability. So regardless of these hypotheses and the results, we believe that there are three main major contributions of this work. First, it will be the first study examining the differential association between psychopathy phenotypes and interoception using neurophysiological measures. Previous studies have only used self-report studies and behavioral tasks. Secondly, we believe that our findings provide new important insights about the pathways, the etiological pathways of psychopathy, namely the trait fearlessness pathway. And finally, we think that this contribution is valid for the field of interoceptive predictive processing to explore the neurobiology of personality. Thank you so much. I also don't have a cool photo to show you in this presentation.
Carlos, and thank you for all the speakers. Were extremely well behaved and practiced their presentations to the minute. So it makes our job of moderating that much easier. Okay, so in these final 10 minutes now we have some time for questions on the talks.
Can I ask Carlos a question? I was just curious because I saw that the HEP, ERP that you're looking at lies within the same window as P300, which is an attentional ERP as well. How are you going to differentiate if the effect that you're seeing there isn't related purely to a cognitive effect, like an attention effect, rather than a cardiac related effect?
Yeah, that's a very good question. And that was the reasoning for us to try to add this modification on the task, because the original task by Petschner, they only did these like blocks where participants have to either count their heartbeats or focus on their heartbeats versus listening to the white noise. And they found this modulation. They actually found on several time windows. And they have like extra analyses that are still not published, but they may be published with like several data-driven methods to try to show that this is consistent. But when we are looking at this task, there was this debate because like counting our heartbeat, it's much more demanding than simply listening to the white noise. So to try to match the task difficulty, and this will be controlled in the pilot study, we'll try to like the white noise has these very slight volume increases that participants have to count. The idea is that the volume increase can match the task difficulty of counting the heartbeat or focusing on the heartbeat. And that way both
act as an oddball.
Yeah,
sort of pattern.
Kind of, yeah. And then we could like have a match in the cognitive effort of the task imagine that if we don't have this manipulation, then like the original task results may be questioned, because now we try to match the the cognitive effort, and then we don't have this manipulation. But yeah, that's why we try to adapt the task because of the P300.
Thank you.
May I ask Katerina a question? Hi, thank you very much. You all did wonderful talks. I just had a question about the mothers that you're getting to come in. So is there a reason why it's mothers and not fathers, or a mix of the two? And secondly, would you expect there to be a difference between, I guess, different sexes of the participants versus the parents? And sorry, a third part to that, what their attachment style is to that parent.
Okay, so I'll start from the beginning to give a little bit of contextualization. What happened was, in my master's degree, I was also studying youngsters and psychopathy. And so what happened was I was taking some samples of some youngsters, and a colleague of mine was doing it with women. And so since some mothers accompany their children, she did it with their mothers and I did it with the youngsters. So when we were trying to produce a paper for the masters in the end, we sort of saw that in general, so we don't know the individual specificities of it because we were studying different things and we weren't supposed to correlate them, but then we just started to see that we will be a nice, interesting paper. We saw that in general, in the general results, the higher traits for specific dimensions were reproduced in women, so most of them were the mothers of these children, and in the children. And so we thought, wait, it could be something there, but we can prove it because that wasn't the study that we were going to do. And so I studied some literature, and apparently not only in psychopathy, but also for some psychopathological situations, mothers appear to have a little bit more of an influence than fathers specifically. However, obviously, it's still not proof that it's the entirety of the influence, and I don't think that's possible. But then with constraints with financial and with times and with COVID, we weren't able to do it for the parents, for the mothers, and for instance, for twins, which would be nice, and for people that were adopted or for people that had biological mothers. And so we had to just restrain ourselves to something. And so we decided to start with mothers because in the literature and what we were seeing in the lab already, there appeared to be some results. So some results for similarities between them. I don't know if that answers your specific question about the mothers.
Yes, yeah.
Yeah, okay. About the sex, in the literature, I haven't seen a lot about it and the parents' sex and related to the youngsters. So I wasn't really considering that, but I may add it as a modulated thing, maybe. But in the literature, they don't pay a lot of attention to that, actually, when they compare parents and youngsters in psychopathy, which is still not a lot done. But when they do, they don't focus a lot on that. So I don't think it will have a very specific impact, but I can't tell you for sure because we still don't know in literature. But about parental educational styles, actually, I have a questionnaire about that is going to be used, I think, by a master's student in our lab. So probably I will be helping her with that. She's going to have the mothers and children that I am going to have, and she'll probably use that questionnaire because she's interested in seeing specifically if educational styles of parents have any influence in psychopathic traits in children and youngsters. So that might be something that happens still for the next year, but for my specific task and for my specific project, I wasn't contemplating it totally. But since I'm going to be helping, I might have some results for you if we talk again about that.
Thank you.
Mayuresh, you were...
Thanks, Fernando. Both Katerina and Carlos, really good presentations. My question is for Carlos, and it's sort of a two-part question. I'm curious to know if you can detect the HEP in resting state EEG. The reason I ask is because obviously the link to interoception and self. And the second part of the question is, if it is, then do you think it's related to the default mode network in the brain?
Thank you for the great question. Regarding the resting state, the initial work with the HAPs was with resting state analysis in the 90s. And there are some, regarding like this self-other dissociation, there is a work, I don't remember the first name of the first author, but that explored like this putative hypothesis that having your eyes closed or open may modulate the HAPs. And these could like index more self-focus versus extroceptive focus. So, but this has not been like consistently replicated. I actually try to look for data sets because there must be like already collected data sets with ECG and EEG with eyes closed and eyes open that could like provide some interesting analysis to see these like whether like adding extroceptive information by opening your eyes modulates these HAPs or not. And this is an interesting question. Regarding like the sources of the HAP and whether this can be linked to specific like resting state networks of that you typically identified in neuroimaging studies, I did not read anything like major on this. I did not find anything major on this. There are some studies like also trying to explore the role of the salience network because of the insula. And I could say like from my standpoint, it can also play a role, but from my knowledge, there's not a lot of information integrating these HAPs and neuroimaging studies, but that will be quite interesting, of course. And this this field in interoception is growing quite fast. I was in a pre-conference last week at the Society of Affective Science, and like it has been growing very fast, which is good and bad because there's also some problems with the tasks that we are using that some people are not being able to replicate the findings. So I think this is like a critical time in the field of interoception.
Thank you for answering that. I mean, obviously, it sounds like an interesting project to do with simultaneous EEG and fMRI.
Yeah.
We need a cable.
Yeah. I actually had another question for Carlos. Sorry for overwhelming you, but will you be measuring any kind of respiration rate at the same time as your heart rate? Just because if you pay attention to your heart rate, you might accidentally end up slowing or speeding up respiration, which could have some flow-on effects, but perhaps isn't really an issue either.
Yeah, that's an interesting question. So the group where I did my training at Munich, in their studies, in the first studies they collected these respiratory data to conduct control analysis. But then, as they did not find like a significant modulation or association in any of the analysis, they actually stopped collecting using this data. I had the intention to use, but like we are using an EEG system that possibly we cannot add these extra channels. I mean, we can add them, but like the time constraints, and because it's like my last year of the PhD, will take too much time to get this set up. But that's an interesting question because in the first studies, like lots of people were controlling this respiratory rate. But now, like the group in Germany did not found any significant association, so I feel kind of safe not using it. Thank you for the question.
Covered, yeah.
Elizabeth?
Yeah, I just had a similar question to Christy for Katerina. In terms of your participants, I don't know if you mentioned it, were you going to look at gender as a difference in the actual adolescence and also the age? Because there's studies that have found like psychopathic traits that are different across ages and different between males and females in adolescence. So I was just wondering how you're going to handle that.
Yeah, so the idea for doing this from the 12 to 16 was trying to understand a little bit if we see differences. So in the age, of course, it's going to be a modulator, because it's important to understand we couldn't do a longitudinal study. And so we decided to have this age gap in here so that we could study a little bit if there was differences, if we could see, for instance, higher traits in specific ages, so like higher in the 12, but then 16 lower, trying to see how this worked, and then of course with the mothers as well. And for gender, what I was thinking of doing with your suggestions now was maybe consider it as a modulator as well, because I wasn't considering it specifically to study. I was more thinking of, well, if we see that we can do it and consider it as a modulator, like see if there's something here, okay? So it wasn't specific for the project, but I think that your suggestions are good. And since already 2 people asked me about that, I think it could be good for me to put it in my project and to try and see if there's any difference, and trying to see also if there's any relation with it to the psychopathic traits. It could be interesting to see. So I thank you for the suggestion.
So what's your sample size going to look like if you're looking for differences from 12 to 16, but in the different age groups?
So we are trying to recruit as much as possible, as many as possible from each of the age groups, so 12, 13, 14, 16, okay? But what is happening, and with COVID, it was really complicated, is that we are not having a lot of people volunteering for these tasks. And so what we are trying to do is to involve the community in here, because that was always a point for me, that scientific investigation should involve community and not only to scientific investigators by themselves. And so what we are doing now is trying to reach out to schools and see if they want to participate with us, if there is something to gain from them, and if they would like their students to participate, and if the students would like to participate. So sadly, we're a bit restrained to how many students want to participate from each of the age groups. So I don't think I'll be able to see exactly what I wanted to see in the beginning of the project, but I'm still going to try and see if I can have at least similar groups for each age, and try to see if I can see any differences there. But with this situation that happened right at the beginning of my PhD project, it's becoming harder to try and do it, but I think we could still find something.
I think we're all in the same boat in that sense.
Yeah.
So we're slightly over time, but I would ask just a couple more minutes of your time. I have a small comment for Sabina, more of a comment really than a question. So I think it's really excellent that you've tackled 2 diseases at the same time, 2 categories at the same time, and I think that's very, very informative, because it there's overlap, but there's also what's specific to each of them. And in your one of your goals is to contribute to help tease them apart. And of course, I mean, to me, this resonates a lot with these relatively recent approaches, like the hierarchical taxonomy for psychopathology. I mean, I'm sure you're probably aware of this. I'm just going to share the big slide they usually show, right? So we start off with signs and symptoms down here. And they're all fuzzy, right? They're all blended together. And then we try to categorize them. And of course, we see that mood disorders are quite close to each other in symptom space, right? So there's a lot of overlap. But then if we try to empirically associate these, and this is what the high top effort has been trying to do, to create a sort of empirical taxonomy for these conditions, indeed, you see distress-related disorders and, of course, mania. They're very close together, but they also have different aspects. And, of course, mania will share some aspects with schizophrenia and other thought disorders. So I think this kind of research is really important, not just, like Rita's work and Elizabeth's work showing how a specific disorder is correlated to brain abnormalities. But if you look at two disorders at the same time, we get this added level of complexity, but also this added level of inferences we can perhaps make about what these systems are how these systems are working across these different symptoms and these different spectrum. So just congratulations on tackling that huge problem that we have of dealing with these categories, but at the same time, we're never sure if these are perfect categories or if we need to revise them in the near future. So that was just a small comment on that.
Thank you. I really appreciate that. Thank you.
Okay, given time, I would perhaps suggest we leave any questions for, we could say the coffee break or the social part of the event. And of course we can do that, but it has to be virtual, right? I'm sure we can all exchange contacts if needed. You can reach out to me or to anyone in the lab. And that's, we're also, I think many of us are on Twitter, so that's also a good means of communication. And just a final reminder of our e-posters. So we have 3 e-posters up on the Padlet website. So be sure to check them out. It's only 3, so it's going to be quick, right? You can do it in a quick break. And if you want, you can leave some questions as a comment. And poster authors, I would kindly ask them to come back in a couple of days and respond to these questions or get in touch if that makes sense. And apart from this, I would just like to thank everyone involved in putting this together, and for the presence, the speakers, of course. You provided with the most important material of all, the intellectual contents, and to everyone here as well. Thank you all for making this possible throughout this huge time zone difference, which is it's really a challenge. But
can I just quickly add, I hope this is the first of many crosstalk between the 2 labs, and hopefully this is a starting point. When we finally are able to travel again, our paths may cross in conferences, and you'll see the familiar face between the 2 labs, hopefully, and chat more about your work. Thank you all for your talks.
Thank you, Fernando, for the idea, and the other Professor Fernando, who's not here as well.
Yeah, he had some teaching responsibilities. He had to rush off.
But it was good to see him. It's really cool to hear research with a Porto accent. And thank you everyone for joining.
It's great as well. Right. Okay.
It was nice seeing everyone and knowing the new faces. I'm very, very happy. This is a very, very special connection for me. So I'm very happy that everyone could hear everyone's work, and we are now all interconnected. And as Mayuresh said, we can start nice collaborations from here, hopefully. Yeah.
And if you want to visit, you're more than welcome. Well, there will be jet lag, but it's to be expected. Okay. So again, thanks everyone, and I think we're now at closing time.
Thank you so much.
Thank you.
Thank you. Have a nice day. Bye.
Thank you.
Bye, Sabrina.
Bye. Bye, Cheryl. Bye. Bye, seeing you all.